Hi guys, how are you? Welcome to my channel. And today's I talk to introduction of microbiology. Let's start. Introduction of microbiology. Microbiology is the study of organism or agent. Too small to be seen with naked eye. It includes bacteria, archaea, virus, fungi, and algae, etc. Microbiology not seen naked eye. It used microscope, and they are present air, soil, and water. And this picture is some shape of bacteria, and this is and this is like some types of virus. And next, and father of microbiology. Anthony van Leeuwenhoek called as father of microbiology because he first using a single lens microscope made by himself, and he first observed the bacteria and microorganism, or he described the first organism he saw as animalcules. Means Anthony Leeuwenhoek called the father of microbiology, and he discovered uh, discovered animalcules, and he uh, made the single lens microscope. And presently modern microbiologists call Louis Pasteur because he first coined term microbiology for the study of organism of microscope size, and he also coined the term vaccine. And this picture is some scientists. First is Robert Hooke, and second is Anthony Leeuwenhoek, and third is Louis Pasteur, and fourth is Robert Koch, fifth is Lord Joseph Lister, next Edward Jenner, next Paul Ansri, and third is Alexander Fleming. Next is Alexander Fleming, and Alexander Fleming discovered the first penicillin. You know. Next, branch of microbiology. Microbiology has two branches. One is on the basis of application, and other is on the basis of taxonomic character. And firstly, on the basis of application include water, air, soil microbiology, food microbiology, etc. And other is on the basis of taxonomic characters, and it include bacteriology, mycology, phycology, and virology, etc. History of microbiology. Microbiology as a science, and uh, discovered the cell first Robert Hooke, and cell theory discovered the Swam and Francisco Reddy and rotten meat, and these are spontaneous generation. And golden age of microbiology is a means fermentation and pasteurization and germ theory of disease. These are golden age of microbiology. And development in the 20th century have discovered of virus and molecular biology and DNA technology, etc. Next slide. Theory of spontaneous generation. Theory of spontaneous generation means living organism can originate from non-living source. Example: rotten meat and maggot. And this picture first is uh, first flask is unsealed, and it has some maggot and meat, and next is flask is sealed, then third is flask covered with geese, and this process is a rotten meat and maggot. or this process is called spontaneous generation. And microbiology diversity. And microbiology diversity can be defined as the range of different kinds of unicellular organisms, bacteria, archaea, protists, and fungi, and the connection between bacteria, archaea, and eukaryotic organisms. The tree of life means archaea, bacteria, and eukarya. Next, Pasteur experiment or spontaneous generation. First, first boil the broth, and broth is a liquid media. Next, broth remaining free of microorganism. Then next, he covered neck is removed. Then he observed microorganism grow in broth, and this procedure is called pasteurization posterior. Next, phylogenetic tree. Phylogenetic tree means microbial phylogenetic is the study of manner in which various groups of microorganisms are generally related, and they are two rotate and unrotate. Uh, rotate means uh, this picture is rotate and this picture is unrotate. Means A species A and species B, both are root is the common ancestor A and B, and this ancestor is common root this, and next species C is a common ancestor this point, and and this point, this blue color point and this blue color point is a common ancestor, and this group species B and species C is not a common ancestor. For example, our parents, uh, my fa father Sil and my uncle Sil not a common ancestor, and my father and my uncle's father means grandfather is common ancestor. For the example, family is a rooted मतलब phylogenetic tree का एक example होगा, and unrooted means not rooted, मतलब इसका कोई रूट नहीं है, कौन सा रूट होगा वो मालूम नहीं होगा. Next slide. Tree in molecular diversity, and there are three types of tree: dendrogram, cleredogram, and phylogram. Classification of microbiology. There are three types of classification. They are five kingdom, next is eight kingdom, and last is three kingdom. And this picture is a some media, some culture media. Five kingdom system of classification. 1969, Robert Whittaker introduced the five kingdom of classification, and this is a Whittaker classification. First is kingdom Monera, second fungi, third animalia, fourth plenty, and last is protista. These are Whittaker 5 classification. Next, 8 kingdom system of classification. Caviller Smith classify this kingdom, and he divided all organisms into two empires. One is bacteria and other is eukaryotia. This diagram is a 8 kingdom classification. Three kingdom classification. This three kingdom classification proposed by Gale Host in 1991, and they divided into three domain classification system: bacteria, archaea and eukarya. And these are difference between archaea, eukarya, and bacteria. Next, method of isolation in pure culture. Uh, the process of scanning a pure culture by separating one type of microbes from a mixture is called isolation. Some common isolation methods are strict plate method, serial dilution, and some example is PDA. PDA means potato dextro agar. Next, strict plate method. It is used for the isolation into pure culture of the organism for a mixed population. Sterile the inoculating needles by flame to make red hot and allow it to cool of 30 seconds. And this process is strict plate method. Firstly, we have peptidase. Next, inoculum is hot and pick the bacteria and first it's a stick line this process. Next, same process again hot this process. Next, this process. This is a strict plate method process. Next, serial dilution. Serial dilution is a series of sequential dilution used to reduce a dense culture of cells to a more usable concentration. Each dilution will reduce the concentration of bacteria by a specific amount. It is used by chemistry, pharmaceutical laboratories, homeopathy, and microbiology. This is a liquid media broth. This is test tube. This process is serial dilution process. Next, serial dilution method has a DF formula. Dilution factor means this formula is volume of sample by volume of sample plus volume of diluent. This and for example, first first test tube dilute factor is 10 to the power minus 1 and second tube dilution factor is 10 to the power minus 1 means 1 ml add to 9 ml and total dilution factor is previous dilution and the next dilution of next tube, so the dilution is 10 to the power minus 2. I write this topic very simple. And culture media. Culture media is substance and food nutrition for the growth of microorganisms. Example PDA, potato dextro agar. And composition of culture media: water, energy source, carbon source, and nitrogen source, and mineral salt. These are composition of culture media. Next, types of culture media. There are very types of culture media and they are divided into two bases. First basis is their consistency and second basis is the ingredient. Based on their constitution include soil media, solid media, uh, liquid media, and semi-solid media. Solid media agar and liquid media broth. Next is ingredients include simple medium, complex medium, synthetic or defined medium, and special media. Next, these are some example. Solid media is a example nutrient agar, blood agar, and semi-solid media, molality medium, and liquid medium is a nutrient broth. Thank you. I hope um, I hope you I hope um, mm, Thank you. Like and subscribe my channel.